Well, good morning, everyone. My name is Riccardo Pressiani, and I'm here this morning with Guido and Matteo, who joined this research project with me. Thank you all for being here. Thank you to the organizing committee for inviting us. It's always, let me say, it's always a pleasure to get these slides back, because it's been a while for the three of us that we've been out of the university. We finished our studies I think more than 1 year ago, I guess. 1 year ago, yeah. And so it's yeah, it's always a nice feeling to talk about the work we've done. So let's get started. Our research work is titled "A Framework for Identifying Group Behavior of Wild Animals." Let me acknowledge the team that worked with us. One of the great things of this research work was that we had the opportunity to work with different universities, different departments, and we had the pleasure of learning a lot from this different perspective of the work we've done. So just to mention a few names, we worked with Roy Arel and Margaret Crawford from UC Davis. They're behavioral biologists. They actually moved to the Max Planck Institute in Germany since a few months ago. Marcus Ambrogio from Politecnico di Milano, where we also did part of our studies. And Tanya Berger-Wolf, who was our research advisor during our time at the University of Illinois, Chicago, and now has moved to Ohio State University. She's the director of the Translational Data Analytics Institute. So just to give you a preview of what we'll be talking about today, we'll start with some context. We'll talk about social individuals, behaviors identification, and so how to identify behaviors in social individuals, and sensors, the sensors that we use to collect data for our research. We'll then go on with the problem definition and the machine learning framework. Guido will talk us about the proposed approach, the data set that we used to train our algorithms, and the results we've obtained. This will mostly conclude what was included in the paper that we published at KDD last summer. But we thought it was a nice idea to give you a bit of perspective on our work, sensor frame. We designed our own hardware to collect for this research. And also, we're going to show you a couple of pictures and tell you about our experience in Kenya during summer 2018, when we went to Africa to test the work that we did and validate our work. So let's start with the context. I'll give the word to Matteo, that is going to introduce us to our research.
Thank you, Riccardo. So in our research, we studied social animals, and we as humans are social animals. Of course, we are not the only ones. So for instance, fishes, dogs, baboon, and birds are social animals too. So at the beginning of our research, we wanted to answer to the question, why do social animals do what they do? So in broader terms, even understanding why we go out on Saturday night or why do we have dinner together. Some animals have some simple behaviors and some social interaction that can be described pretty easily. So for instance, if you consider the school in fishes, when the fish in the center moves to the right, all the other fish will move to the right too, in a synchronized fashion. So this behavior can be described with a simple mathematical model. However, that is not the case for more complex animals. So for instance, in our research, we studied baboons, which are social primates. So if you look at this video, you'll see that baboons perform many more activities. So you will see a baboon in the center, which is carrying a baby, some baboons that are foraging, and some others that are having other interactions. So these behaviors cannot be described with unique mathematical models. For this behavior, you will need more complex tools such as machine learning tools. So let's go to the problem definition and start to understand the problem from a biological perspective. So we study social individuals, and again, we study social primates and specifically baboons. So the problem is that the biologists want to record what the baboons are doing, and they want to do it at different levels and resolution. So even though this might seem a pretty easy task, it's actually pretty complex. In fact, the task is time consuming because the biologists have to look constantly at the baboon for taking notes. And also the number of individuals that a single biologist can track is limited by the resolution of the analysis. So in other words, if you want to track a group of baboons, you won't be able to record what each individual is doing, but just the group level activity. If instead you want to record what each precise individual is doing, you have to focus just on a single individual. So the first solution to this problem is the use of sensors, because the sensors, provided that they have a good battery life, they will be able to constantly monitor the baboons. So the sensors that we use are placed in a collar and a bracelet, and if you have enough collar and bracelet, you will be easily track a higher number of individuals. So you also solve the second issue. Finally, using machine learning, you will be able to understand what animals are doing with a good resolution. So even though the sensors seems the solution to this problem, they actually collect really complex data. So these are the data collected during just one day of data collections. And as you can see, they are multivariate time series. So to understand what this data means, you will need complex machine learning tools. Even to expertise, these data are meaningless otherwise. So looking at the problem, we have as input the behaviors of the baboons. Basically, it's a label telling us what the baboon is doing. Can be running, walking, for instance, or foraging. We have social individuals, which are baboons, and time series collected by our sensors. From a biological perspective, the biologists represent the answer. So in other words, the biologist is able to understand what a baboon is doing and can track the animals, but it has all the limitations that we've seen before. Moving to a computer science perspective, the biologist becomes part of the input, and the machine learning becomes the answer to our problem. So more formally, we have two partitions of data, one with labels and one without labels. And the behavior identification problem is the process of creating a machine learning model able to infer labels for the unlabeled partition using the information in the labeled partition. So in other words, telling us what the baboons are doing after a training on. So now I'll leave the words to Guido that is going to explain you the machine learning framework that we developed.
Thank you, Matteo. So what I'm going to do is that I'm going to explain the step of the learning framework that we developed for this research work. In particular, the framework starts with a label mapping process. As Matteo was describing before, the label provided by the biologists are used as ground truth and as label in this research. So we need to synchronize them with the sensor reading provided by a sensor, providing temporal synchronization method that can synchronize the time of the sensor and the time of the observation of the biologist. Then what we are going to do is to provide an optimal temporal resolution selection algorithm. In that way, for example, you can think about behavior, which, like, for example, running, it's a pretty fast behavior, and it changes in seconds. Whereas, like, for walking, it may change in minutes, and for sleeping behavior, it may even change in hours. So this process will allow to find the global optimum for providing an optimal resolution for doing the classification of the behavior. Then, as we have described before, these animals, and in particular baboon, are social primates. They develop a relationship. So we use network, and in particular social network, as to explicitly encode the relationship between these different animals. Finally, we will have our learning process. We just use a general purpose model, XGBoost, and we provide the feature engineering in the previous two steps and the inference needed for the model to learn the classification of the behavior. So more in depth in this step of the framework, the temporal resolution selection use a general purpose model, as I said before, XGBoost. We try different values that were meaningful given the expertise of the biologist. And as you can see, the model will go through the data, and we have a timestamp, we have the ID of the animals, we have features, and we have labels. So let's say that this black box that you see in the structure of the dataset is the right resolution. What we are going to do is that we are going to aggregate all the data reading from the sensor given this temporal resolution, creating time windows, and the label will be the majority label of that temporal frame, because that is meaningful from a biological perspective. Then we are going to infer the network to explicitly encode the social relation. And to do that, we need to select the node. So for the node, it's pretty easy. We have an ID in the dataset, and we just use the ID as the node. So we have different baboon, as you can see in the slide, with different colors. And then we use the feature. So many of these datasets based on sensor and activity readings contains latitude and longitude from the GPS. So we just use that, and we infer proximity network for each time window that we have inferred at the previous step. As you can see, we have a proximity network for T1, and we have that for all the time series that we have extracted before. Finally, given this network, we, as I said before, explicitly encode the relational features and the structural features, like, for example, how many baboons are connected with me, or like what the baboons near me are doing, because our assumption is that the neighborhood of an animal define the behavior of that animal. And these assumptions are also based on literature reading in the biological literature. So this is the final data set that we got after this step. As you can see, we have aggregation feature and temporal features coming from the temporal resolution selection. And then we have structural features and relational feature coming from the network inference. The data set where we evaluated our work was collected in 2012 at the Impala Research Center in Kenya. It was the first time ever a complete group of social primates was tracked with a GPS and an accelerometer collar. And the data set resulted in 35 days of complete observation on 26 social individuals. And this data set is also publicly available if you want to look at it and do some insight also in this activity problem. We have 12 hours per day sensor reading, and the label provided by the biologist was a social group label. In particular, we have 8 different classes and about 20 hours over these 35 days on ground truth. And I just wanted you to focus on the frequency of the label provided by the biologist. As you can see, basically, if we consider the first 3, which are progression and like a general hanging out label, they cover 90% of the ground truths available in this dataset. So many models and many in the literature just consider the majority class without respect to the lower represented class. But in this research, we explicitly focus also on the minority class, and we will provide metric to measure that our proposed approach is able to actually classify all the behavior and not only the more represented. So more formally, the dataset, as Matteo was saying before, is composed by time series. We have a time series, a multivariate time series for a baboon. And we encode this time series also for all the other baboon. And we may have different labels for baboons at different timestamps. And our general purpose model will be able to actually apply that circle color that you are seeing in this slide for all the 35 of observation. So the evaluation metrics that we consider are pretty standard. For example, the accuracy will allow a comparison between our method and past method, and also our method and new method that will come from future research. But also, as I was saying before, the F1 measure will provide us the metric for understanding that we are actually able to classify all represented class, and we are going to consider both micro and micro and macro average for the F1. Then pretty quick about the baseline, which we compare our model. We took from the activity recognition literature, and in particular from 2 main fields, sequence analysis, from which the method that I just presented is part of, and deep learning. And in particular, we consider the AST, which was the best method on this explicit dataset before this proposed solution, and deep conval STM, which has been proven to be one of the best deep learning methods for activity recognition. Also, as the dataset is highly unbalanced, we provide the majority classifier result because it will still be significant with respect to our method. And so we really want to understand if our method is able to provide a better classification than just apply the majority activity. So a quick view about the result of the temporal resolution selection. We tried 3 different methods: 1 minute, 2 minutes, and 3 minutes. And as you can see, 1 minute is the best value. You may think that aggregating 1 minute instead of 2 will provide a bigger data set for training. So we explicitly sample the same number of training instances for each of these values. In this way, we can have a fair evaluation of all these different temporal resolution values. And after that, we show the time series to our biologists. And as you can see, this is Margaret Crawford in the slide, and they always approve our results also from a biological perspective. Then the final result of our approach are presented in this slide. As you can see, we are almost 10% higher the best baseline for both the metrics. And this is due especially to the explicit representation of the social relationship of this baboon, which were not encoded, not in the AST model and even in deep conval STM. And also these results have been biologically approved by the biologists working with us. And the way I'm saying biologists approve when you're seeing these green boxes is because we made videos about the prediction that we made in the dataset. So as you can see, these are 3 individuals in Kenya from the dataset in 2012. And what is happening is that the red will represent stationary behavior, and the green one will represent a non-stationary behavior. And as you can see, the prediction is quite correct, and the biologists were using this to understand if they could use our labels for doing their biological analysis. So summarizing the contribution that this work and the KDD publication had, first of all, we developed a method for finding an optimal temporal resolution selection and an optimal temporal resolution value. Furthermore, we provided a method for direct encoding of social relationship within a group of social animal with a social network. And finally, we evaluated our approach on a real-world data set, providing insight and showing that our method outperformed the current state of the art. So given that, as Riccardo was saying before, we also move forward to continuously improve this research and this work. And Riccardo will now talk about the sensor framework that we developed in our Kenya trip.
Thank you, Guido. One of the, in my opinion, one of the greatest aspects of this project was the continuous loop that we created during the development of the machine learning framework and the development of the sensor. What I mean by that is that when we built the sensor devices, we thought about how to collect the most meaningful data for the machine learning. But then when we worked on the machine learning framework, we find out something that could be changed in sensor devices to improve the results of the machine learning. And so we went on and on with this process to make both of the parts of this project better. So when we thought about building a new device to collect data, we obviously looked at the state of the art. And at that time, the e-obs color was one of the mostly more recognized devices to do this kind of research. You may also have heard of this device. It's pretty light, is small, and regarding sensors, it's equipped with a GPS module, a 3-axis accelerometer, and a VHF radio link to dump the data while the device is deployed on the animals. This is exactly the same device that was used to collect and build the data set that Guido mentioned before, that we used to train our algorithms. So after looking at the state of the art, we then stopped and think about what should have been improved and what we should have done to get the most out of the data collection. So among the requirements that we got, I'm going to mention a few of them. We wanted a broader set of behaviors to be recognized with the data collected, and I'm going to talk more about that later on. We wanted to enable micro-localization, which is a more granular localization rather than GPS. Of course, we wanted the device to be fault tolerant because we couldn't just recapture the animal whenever something could have happened to the device. For the same reason, we wanted to allow remote data retrieval, and we wanted a decent battery lifetime to allow a good time period of data collection. Also, these devices were also thought with the animals in mind. As you might think, there are a large number of issues and potential problems when you develop a device that has to be deployed on animals. And so we had to take into account all these problems. In this presentation, we're going to focus on, mainly focus on 3 of these requirements. The first one is the broader set of behaviors. The picture you see on the right is an ethogram. The ethogram is a structure that biologists use to categorize and create a hierarchy of the behaviors of an animal species. And in particular, with the existing device, we were able to detect activities such as sitting, walking, and basically most of them related to the movement of the animal with respect to a map. However, the goal of our research was to detect many more behaviors. So for example, in the individual category, we wanted to detect behaviors such as foraging, which needs information about the movement of the arms of the animal. And also the focus was on 2 other categories of movement, such as dyadic behaviors and group behaviors. When I mentioned micro-localization before, our idea of micro-localization versus macro-localization is that with macro-localization, you are able to place an animal using the GPS data on a map. With micro-localization, what we wanted to do was to position the animals with respect to each other, so that we could have been able to recreate the social network of the smaller groups inside the larger one. Also, we wanted to implement remote data retrieval, as mentioned before, and in particular, a 2-way communication between the devices and a hub. So the idea was to being able to tune the operational parameters of the device during the deployment without having to collect the devices back and every time we needed to do that, but also to collect the data before retrieving the devices. As a matter of fact, the case of our devices was equipped with an automatic unlocking system, so that we didn't have to recapture the animal to get the devices back and also the data. This is the overall system architecture of our sensor system. As you can see, we have 3 main parts. The first one is the bracelet, which allowed us to get information about the movement of at least one of the arms of the animal. And so it enabled us to make the set of behaviors recognizable by the software broader. The bracelet communicates through Bluetooth Low Energy with a collar. And the same Bluetooth module implements a micro-localization system, a simple proximity sensor. Finally, the color communicates through Wi-Fi with a hub, making us able to download the data during the experiment. Here you can see a picture of the board we prototyped. This in particular is the bracelet. As you can see, it's pretty small. And in terms of sensors, we have a 6-axis inertial movement unit, which is composed by an accelerometer and a gyroscope. We have a real-time clock and a controller. These are the pictures of the 2 faces of the board of the collar. We have a real-time clock as well on the collar, a microphone, a 9-degree of freedom inertial movement unit, which has, again, an accelerometer and gyroscope, and also a magnetometer to understand the direction the animal is facing. We have 2 controllers, a GPS module, and an SD card to store all the data collected. Just a few words about how the 2 devices, the bracelet and the collar, works from a software perspective. The bracelet has 2 phases, a setup phase where the whole system is bootstrapped. And then the bracelet goes into a loop phase where it continuously samples the data and send them to the collar through Bluetooth. The life cycle of the collar is defined by 4 states. Similarly to the bracelet, we have a boot phase where the whole system is started. And then the system goes in a collection mode where it collects data. And after the collection mode is completed, it goes in a Wi-Fi state where it makes the device available to connect to the hub so that we can download the data and then restart the collection phase. We did some experiments, some tests of the features that we implemented in our devices. And the first one was the proximity sensor. The idea with the proximity sensor was to address the distance less than 5 meters, below the 5 meters threshold, which is exactly the range where the GPS start to lower the accuracy. We were able to obtain pretty good results. As you can see, we can distinguish pretty clearly 4 interval of distances. And this was what we needed to recreate the network of the individuals in smaller groups, very close to each other. Then we tested the GPS. When we were in Kenya, we built this triangular structure. And the goal of this experiment was to make sure that a set of devices deployed on animals would have continuously kept the relative position among themselves during time. And so, as you can see in this video, the 3 dots continuously move toward the right, but always keeping the relative position back to each other. We calculated an average error for the GPS data of about 1.2 meters. Always in Kenya, we tested the Wi-Fi connection of the collar with the hub. And of course, we have to keep in mind that we were in Kenya, and so it was a very noise-free environment without basically any interference in terms of radio signals. And so we were able to establish a connection between the hub and the devices at a distance of about 100 meters. Lastly, we tested the battery lifetime of the bracelet. And in particular, this information is critical for the bracelet because even more than the collar, we had to be careful about the weight. We didn't want to interfere with the daily activities of the animals, and so we had to make it as light as possible. With the small battery that we placed in the bracelet, we were able to obtain a battery lifetime of about 80 hours. And if you think about collecting data for 12 hours per day, so basically stopping the data collection while the animals are sleeping, we can get to almost 1 week of data collection. To conclude, we just wanted to spend a few words about our experience in Kenya during summer 2018. We flew from Chicago to Nairobi in Kenya. We then drove to the Impala Research Center, where we met with a team of biologists. Our goal there was to test the hardware that we developed. We wanted to design a protocol for data collection. Guido mentioned that having a part of the data labeled by the biologist is needed to train our algorithms. And so we wanted to go in the exactly same place where the device is deployed, so that we could see with our eyes real close the baboons, see how they behave during the day, and what is the collection protocol for the biologist, and how we could adapt it to our purposes. The idea then was to deploy the system on, and I'm saying that this was the idea because unfortunately, a couple of days before leaving to Kenya, the Kenyan government raised a moratorium on animal experimentation, and in particular, the problem was with the sedation of animals, and we couldn't deploy our devices without sedating the baboons. So we couldn't find any way to get an authorization to do that. And so instead of deploying on baboons, we had to revert to sheep. And lastly, we wanted to validate the machine learning framework in the field. So here's a couple of pictures about our experience. We went there with the biologists from UC Davis. You can see in the picture on the right the cases of the devices. We put them together and we make them work. We then designed the protocol collection. We went out in the field to see how the biologists work, and we then draw out on that whiteboard all the behaviors that potentially we wanted to detect from the data we collected. Here you can see a couple of pictures of the deployment we did with sheep. You can see our devices in the picture on the top right corner. And even if the behaviors that we can see from a sheep are much simpler than the behaviors of a baboons, we were able to collect data to identify 4 actions, 4 activities: standing down, standing up, and walking down. And we were able to predict these activities with a pretty high accuracy. I think our experience in Kenya was fantastic. We had the unique opportunity to go in this environment in the middle of nowhere, where all we could see was nature and the animals in their real environment. So this was also a great addition to our research. Here's the picture of the complete team that went to Kenya. And to conclude, we were happy, pleasure to present our work in Alaska last summer at KDD. Guido went there with Tanya, our research advisor. And this is the paper that you may have looked at. Well, this concludes our presentation. Thank you very much for your attention. Again, my name is Riccardo Pressiani, and I'm here with Matteo Foglio and Guido Muscioni. Me and Matteo co-founded a software consulting company after graduating, and trying to apply all of our experience and expertise that we developed during our time at the university in the work we do. Guido instead is working for Anthem. He's an AI engineer there. And right, so we'll be here for a while. So if you have any question, we'll be happy to answer them. Thank you.
Could you talk a little bit about what WePo does?
Excuse me, could you repeat that?
I was saying, could you talk a little bit about what WePo does?
Our company, yeah. We're a software consulting company. We're now trying to apply, we focus on AI and machine learning methods for other companies that may need our expertise. We're still talking with a couple of potential projects to apply this same work that we did during the university. Hopefully we'll have the chance of working again in this field because we really loved it. As of now, we're applying our machine learning skills in several projects. Some of them are related to extract information from documents of any type. So feel free to contact us. I don't know if we left our contacts anywhere, but I'll make sure to send a couple of links in the chat so that you can look up our projects and possibly get in touch with us if you want to know more.
Thanks.
Hi, I have a quick question. In the first part of the talk, you referenced the KDD paper. Would you mind sending out the link for that? Or if you give us the title, then I can go ahead and Google it.
I think we can get the link and post it in the chat.
Sure, thanks.
Hi. So I was just curious. So you say that like in general, your company isn't working as much like in the field. Sometimes it might just be other random machine learning and AI projects. So it seems like a lot of the work you had to do with localization and hardware, was that like separate from a lot of the other projects that you will work on? And if so, like how was that very difficult for you in terms of like a learning curve to get all of that infrastructure set up with everything involved with the GPS and the proximity sensing and the localization?
Well, the work we presented we developed is basically our research activity that we've done while in university. Okay. So everything that we basically WePo and the company is a whole after we graduated, and so I'd say the learning curve was during our time during university, and I mean I can say that everything was very useful to what we're doing now. So I think I can say that the work we did with this project we presented was kind of the training for us to then go out and starting to work with clients and possibly in different fields.
Okay.
Yeah, right now we're working mainly on machine learning and not anymore on hardware. And we're working on different fields like from governmental data, finance data, or also tracking behavior, but in this case of human.
Okay, cool. Thanks.
Does anyone have any other questions?
Yeah, I guess if no one else says, I have some more questions. So like with, you said you did some work on figuring out like the optimal time resolution for your frames. So what was the optimal time resolution, or was there multiple resolutions that were optimal?
So I can take this video. So basically we tried 3 different values as I have shown before during the evaluation of the machine learning framework. We tried 1 minute, 2 minutes, and 3 minutes because these were meaningful for the group behavior label that we were considering, but our method is free from that parameter, and basically providing a set of different temporal resolution, it will be selected. The best one will still be selected based on the metric that the user can provide on, in the algorithm.
Okay. Another question I have is like, is this biology group that you worked with, are they going to continue to use the sensors and algorithms that you created for them, or was it like a temporary thing?
So regarding the machine learning framework, we provided and we labeled for them all the 35 days of observation for the social group behavior. And we also provided individual behavior, where part of the dataset were not published at KDD, but we also developed a more accurate pipeline for individual behavior. And they are currently using our prediction for taking conclusion on how these baboon lives in the wild, how they structure their society and the hierarchy between them. And there are some interesting takeaway from them. For example, like even if baboon have a leader, they usually tend to follow a majority rule, and the group moves with respect to the majority direction of the group instead of following the leader. And so much more insight, they were able to get so much more insight given the fact that they had 5 days of full behaviors.
How many days of that full behavior were human labeled for your training and also for your evaluation?
So for the social group behavior, which is the one that we presented today, we have about 20 hours of ground truth, which means almost 2 days of observation because we have 12 hours per day. Regarding the individual behavior that I described before, we had 2 hours within all the 35 days of activity. So a pretty low, like a pretty small set of ground truths to provide the full prediction of the other days.
Sorry, so you said you have 2 days for the training and 2 hours for the evaluation?
No. Oh, you're saying like what was the structure of the evaluation, training, and testing splits?
Yeah, just like how much data do you have for training, and then how much data do you have for evaluating the accuracy of your classifiers?
Okay, so basically we always applied standard technique in the literature for evaluating prediction. We basically always use in the social group behavior, as I said, I had 2 days. So we train it on 1 day and classify on the other day.
Okay.
And then we train it on the second day and classify the day in the past, just to provide a more view on the evaluation of the method.
Okay, and what were your accuracies for your evaluation?
So we, as we were showing before, these are the accuracy of our work, so 77%.
Okay. Right. And that was on a like for some set of behaviors that you were able to test on the sheep?
No, this was for the data set of the baboon. Regarding the test that we did on sheep, we did the same split. So for sheep, we collected 2 days of data when we were in Kenya in 2018. And then also in this case, we classified on the first day and predicted on the second day. And as you can see here, the accuracy is much higher. This was due to the fact that, as Riccardo was saying, the set of sensors is richer. And so we have much more data. And also the behavior of sheep are pretty, are much more simple compared to the.
Okay, yeah, I understand. Thank you.
Any more questions? So if not, let's thank our presenters today. And it was a really great presentation.